Today, we are going to talk about plant hormone ethylene. We'll discuss its structure, discovery, and biosynthesis. Ethylene is the only plant growth regulator that occurs in the form of a gas and one of the first plant hormone discovered. It is a volatile gas present in the atmosphere as a component of smoke and other industrial gases. Ethylene is produced by almost all parts of higher plants and usually present in minute quantity but causes marked effects. Like abscisic acid, ethylene is usually considered as inhibitory hormone. It is a colorless flammable gas denoted by formula C2H4 having sweet and musky odor when pure and unsaturated hydrocarbon having double covalent bonds between and adjacent to carbon atoms. Being volatile in nature, ethylene gas is not used directly. Commercially, it is being used in the form of a substance ethylene, 2-chloroethyl phosphoric acid, which after hydrolysis yields ethylene gas. Discovery of ethylene. Ethylene has been used since the ancient times. Egyptian gashed the figs in order to stimulate ripening. The ancient Chinese would burn incense in closed rooms to enhance the ripening of pears. In 1864, it was discovered that gas leaks from street lights led to stunning of growth, twisting of plants, and abnormal thickening of stems. In 1874, it was discovered that smoke caused pineapple fields to bloom. In 1901, a Russian scientist, Dimitry Neljubow, observed that dark-grown pea seedlings growing in the laboratory exhibited symptoms like inhibition of stem elongation, stem thickening, and horizontal growth. When the plants were allowed to grow in fresh air, they regained their normal morphology and rate of growth. He identified it as a ethylene, which was present in the laboratory air from coal gas as a molecule causing the response. In 1912, A.F. Seward and R.H. True demonstrated that the combustion gases from the kerosene stove were beneficial in the ripening of lemons. W.C. Croker and L.I. Knight in 1908 and 1913 respectively identified ethylene as the active constituent of both illuminating gas and tobacco smoke. The first report that plant materials evolve ethylene came from H.H. Cousins, who observed that when oranges and banana were stored together during shipping, some gas was emanated from oranges which caused ripening of banana. Sarah Dowd in 1917 discovered that ethylene stimulated abscission. R. Ganney in 1934 provided a chemical proof that ethylene was produced by ripe apples. Since then, it has been shown that ethylene is produced from essentially all parts of higher plants, right? Next is biosynthesis of ethylene. The pathway for ethylene biosynthesis, also called Yang cycle, and is given by its Gang for Yang in 1980. First of all, the sulfur containing amino acid methionine, this is methionine, it is activated with ATP and is converted to S-adenosyl methionine, right? To means SM, and pyrophosphate is liberated. And this reaction is catalyzed by the enzyme SAM synthetase, this enzyme. In S-adenosyl methionine, the sulfur atom of methionine is linked to carbon fifth of the ribose moiety in adenosine. In many cases, selenomethionine has been found to be the better substrate for ethylene biosynthesis than methionine because enzyme SAM synthetase has the higher affinity for selenomethionine than for methionine. The next step in the biosynthesis of ethylene is the conversion of SAM to 1-aminocyclopropane carboxylic acid, ACC. This compound, which is catalyzed by enzyme ACC synthase, ACS, right? This enzyme. This was the first step, and this is the second step. In the final step, here oxidative cleavage occurs of this 1-aminocyclopropane carboxylic acid to form ethylene plus carbon dioxide and HCl by the enzyme ACC oxidase, ACO, right? Formerly, this enzyme was known as the ethylene forming enzyme, EFE. HCl, which is released, is ultimately converted to formic acid and ammonia, and this reaction requires oxygen and is activated by light. And here, this step, this is methionine cycle. This how from this step methionine is produced, right? So this is methionine cycle. Next is physiological role of ethylene. First is fruit ripening, seed germination, then flower inhibition and sex expression, epigenetic responses, then growth inhibition and morphogenetic effect, then acceleration of senescence and abscission. It also regulates the growth of cell wall, stimulates the formation of new roots, induces the growth of adventitious roots during flooding, induces root hair growth, and induces de novo synthesis of peroxidases, right? Thanks for watching. If you like this video, make sure to subscribe for more. Thank you.